Hi everyone, let's talk about the extremely rare Bombay blood group. We're going to start with a mystery. This is a pedigree. Squares represent males and circles represent females. This couple has a daughter and a son. But here comes the interesting part. The father has blood type AB, the mother has blood type B, and their son has blood type A. However, after some blood typing, they found that their daughter's blood type is O. How is this possible? Your first thought might be that one or even both of her parents are not her birth parents. She might be adopted. However, I can assure you that she is really their daughter. To solve this mystery, let's first review what blood types actually mean. You might be most familiar with the ABO system, which is comprised of blood types A, B, AB, and O. Antigens are structures, usually proteins or polypeptides, that are on the surface of cells or pathogens. They are like flags, which act as signals to the cells of your immune system. They are able to communicate whether the structure they are on is produced by your own body, which would be seen as safe, or whether it's foreign. If they're foreign, your body produces an immune response to kill the foreign invader. This is why blood transfusions can only be done if the blood types are compatible. If your body doesn't already have a particular antigen found on a donor red blood cell, the antigen would be recognized as signaling a foreign invader, and an immune response could occur which could have serious consequences. For blood type A, the A antigen is present on the red blood cells. I'm going to draw these antigens as triangles. These people would also have B antibodies circulating in their blood to prevent against foreign red blood cells that have B antigens. On the other hand, blood type B has the B antigen present on their red blood cells, which I'm drawing as squares, and A antibodies in their blood plasma. Blood type AB has both A and B antigens on their red blood cells. This occurs due to them inheriting one A allele from one parent and a B allele from their other parent. The A and B alleles have a co-dominant relationship, so both A and B antigens are going to be expressed in the same person. As these people have both the A and B antigens, those antigens will both be considered safe. So, these people will have neither A nor B antibodies. Those with blood type O is commonly said to have no antigens, but we'll soon see that that's not really the case. People with blood type O will have both A and B antibodies, since both are considered foreign. Genetically, the O allele is recessive to both the A and B alleles, and it's normally written as a lowercase I. You might also know whether you are positive or negative for a blood type. The positive and negative refers to whether you have the Rh factor on the surface of your red blood cells. The Rh factor, or rhesus factor, is another protein that is unrelated to the ABO blood types. Positive means that you have this Rh factor protein on your red blood cells. I'm going to show these as lines. Negative simply means that the Rh factor protein is not present on your red blood cells. I'm mostly explaining this to highlight that the Rh factor does not influence whether you have blood type A, B, AB, or O. The gene that encodes for the ABO antigens and the gene that encodes for the Rh factor antigen are on different chromosomes in your genome, and so they have independent assortment. This gene is also unrelated to the Bombay blood group. So, let's go back to our pedigree and see if we could assign some genotypes. We could assign the father's genotype right away, since we know that AB blood must have one A allele and one B allele to result in the co-dominant phenotype. For blood type B in the mother, there are two possibilities. Either she is homozygous B, meaning that she inherited the B allele from both of her parents, or she is heterozygous, meaning she inherited one B allele and one O allele. We would need to look at their next generation to see if there's anything that might point us to one genotype or the other. Their son's phenotype is blood type A. Since there's only one A allele in the parental generation, he must have inherited the A allele from his dad. This also means that the only way he could present with blood type A phenotype is if he inherited the O allele from his mom, making him a heterozygous and also confirming that his mom is heterozygous for blood type B. If we draw a Punnett square out, now that we have confirmed the genotypes of the parents, we can see the possible phenotypes that their children can have, as well as their predicted likelihoods. The parents have a 25% chance of having a child with blood type AB, 25% chance of blood type A, and 50% chance of a child with blood type B. However, it does seem like it's impossible for them to have a child with blood type O. Well, it turns out that this is epistasis at play. Epistasis refers to interactions between two or more genes. We usually see epistasis when there is a reaction or signaling cascade. One example might be that gene A encodes a protein that is needed to activate the transcription of gene B, and protein B is needed to activate the transcription of gene C to produce protein C. If you happen to have a mutation in gene B such that protein B is not properly produced, it doesn't matter which form of gene C you inherit, because protein C will not be expressed even if you have gene C. Here, it turns out that even though blood type O does not have A or B antigens, it still has a precursor antigen, in this case called the H antigen. So, over here, no antigens should really be in quotations. As you might recall, antigens are often proteins or polysaccharides, which are both chains of their basic structural unit. In this case, the A and B antigens are actually built on top of the H antigen. So here, I've drawn a diagram to explain this. The first block represents the other units that come before even the H antigen. The second block is the unit that's added to create the H antigen. In people with blood type O, this is the basic antigen they have on the surface of their red blood cells. Following the H antigen, another unit can be added on top to create the A or B antigen. But as you can see, without the H block, neither the A nor B block can attach. Therefore, if you happen to not have the H antigen, you won't be able to express the A or B phenotype, even though you have inherited the trait genetically. Again, the gene that codes for the H antigen and the gene that codes for the A and B antigens are on different chromosomes. And this is exactly what happens to people with Bombay blood. People with the H antigen, usually written as an uppercase H, has the precursor, so the expression of the ABO gene can be properly expressed. In people without the H antigen, written as a lowercase h, they don't have the H antigen precursor, which prevents them from expressing the ABO phenotype. The gene that codes for the H antigen has a typical dominant recessive relationship. So, we can see how this pedigree might happen. The daughter over here probably has a homozygous recessive H allele, which prevents her from expressing her ABO phenotype. This means that both of her parents must be heterozygous, as they themselves express AB and B blood types. Her brother, on the other hand, could either be homozygous for the dominant allele or heterozygous like his parents. But a final question remains. Why does she think she is blood type O? Well, you might know that blood typing works by mixing some blood from a blood sample with some antibodies. I have a very basic example here, with one control at the top and two tests at the bottom. The control has no antibodies added, and it's primarily to see if there's any contamination that might cause the blood to clot on its own. If procedures are done correctly, we should see blood samples that don't clot. The second row is added with A antibodies. The two samples on the right have clotted, while the first sample hasn't. Now let's look at the third row, which has B antibodies. And we see that two of them have clotting reactions, while the middle sample doesn't. This shows that the first sample is blood type A, since only the A antibodies recognize the A antigens on the red blood cells, which caused a clotting reaction to occur. For the same reasons, we can deduce that sample 2 is blood type B. However, sample 3 didn't have any clotting reactions here. So that means that neither the A nor B antibodies detected any A or B antigens on the red blood cells in sample 3, making it blood type O. Do you see any problems with this test? By only testing the blood samples against A and B antibodies, the results for sample 3 could also mean that this blood is Bombay. A special H antibody would have to be used to perform a definitive test for Bombay blood. Since the blood type is so rare, most countries usually just test for the ABO system and Rh factor, leaving Bombay blood entirely out of consideration. This type of mix-up can be dangerous. If a person with Bombay blood was given a blood transfusion with type O negative blood, which is commonly regarded as a universal donor, they would still experience an immune response, which could lead to fatal outcomes. This is because of the H antigens that are still on type O blood. That's why Bombay blood can be considered as an even more universal donor than type O blood.